Common ground within. George Lucas thrilled us with his fictional universe in the Star Wars movies. His favorite creation, or my favorite creation of his, were the midichlorians. These were microscopic entities that lived inside the cells of other beings, where they applied a connection to the mystical force. The more midichlorians a being had, the better connection they had with the force. And Darth Vader had the most midichlorians. Even when his body was battered and weak and frail, he still exhibited plenty of power thanks to his midichlorian partners. The midichlorians lived as symbionts within the body of a living being. In fact, they were endosymbionts living within the cells of another being. George Lucas fashioned the midichlorians after the true endosymbiont, which are the mitochondria. Now, the mitochondria are energy-producing organelles that are in your cells. The more mitochondria a cell possesses, the more energy it can produce. Now, the mitochondria themselves have a very interesting story. In the 1960s, a theoretical biologist, Lynn Margulis, proposed that these mitochondria were actually relics of ancient bacteria. They were engulfed by larger cells that shared their space. Inside the larger cell, they were protected from the harsh environment, but they also continued to produce energy. Both cells lived in a symbiosis where they both benefited from this relationship. This landmark paper of Lynn Margulis was rejected by 15 scientific journals before it was finally published in 1967 in the Journal of Theoretical Biology. Since that time, scientists have had decades to provide evidence and support that this idea actually made a lot of sense. In fact, it was elevated to the status of a scientific theory called the Endosymbiotic Theory. Now, we know that a scientific theory is not just a guess, but it's science's best explanation of events. And the Endosymbiotic Theory, proposed by Lynn Margulis, explains the origin of complex cells. I had a great pleasure to work with Lynn Margulis in the 1990s as an editor at W.H. Freeman and Scientific American Books. And the image that you see on the screen here was on the cover of one of her Freeman books called 5 Kingdoms. It's a great image. It was designed by her son, Dorian Sagan. In her youth, Lynn Margulis was married to Carl Sagan. Now, Carl was another scientist, but he was also host of America's most popular public television show, Cosmos. He was associated with the phrase billions and billions of stars, I guess, maybe galaxies. Although he says he never uttered that phrase on Cosmos, he was still honored with a new unit of measurement called the Sagan. The Sagan is just a very large number of anything, at least 4 billion. Well, Carl Sagan taught at my alma mater, Cornell University, when his image appeared on the cover of Time magazine. He was standing in an ocean of stars with clouds around his feet. I remember being invited to a party at Cornell. Everybody showed up with stars and clouds pinned to the legs of their jeans because it was a nerd party. That's the party scientists go to. You have to watch inviting scientists to parties because they bring up random topics like poop. Are alien life forms. Dr. Sagan was also invited to the party, but he said no. It's too bad, though, because I think he would have fit right into the conversation. Tonight I want to talk to you about a different group of symbionts, and that is the trillions of microbial partners that you have right there in your body. In 2007, the National Institutes of Health initiated the Human Microbiome Project, trying to identify all the species with whom you share space or a common ground. These bacteria are found in our skin, they're found in our orifices, they're particularly found in our gut, that is the tube that runs from the mouth to the anus through our body. A microbiome is defined as a community of microbes and their genetic material. Some of the work on the Human Microbiome Project was done at the Genome Institute at Washington University, led by a scientist named George Weinstock. This was his take on the topic. If you think of the human body, it's made up mainly of the microbiome. Then there's the human part of us that provides the environment for all of those microbes to live in. Now, you probably have always thought of yourself as a single, self-sufficient being. Weinstock would argue that you're actually a complex ecosystem made up of your cells and all the microbial partners that you have, cooperating, communicating, competing, coordinating. So what do we know so far about the human microbiome? Well, we know that we're outnumbered. That's really not new information, though, because in 1972, another Missourian, T.D. Luckey, said that there were 10 trillion bacterial cells for every 1 trillion human cells in the body. In fact, his numbers were 100 trillion to 10 trillion. But he got this information by analyzing feces. Still, that number stuck. It was a fact for over 40 years. In 2013, another paper was published by scientists that actually went into the human body, and they looked at the various organs and tried to estimate how many cells there were. They came up with 37 trillion, give or take a few trillion. So the ratio now is considered more like 3 to 1, and yet we're still outnumbered. The important thing is not so much the number of cells, but the number of genes that those cells contribute. Thanks to the Human Genome Project that was finished in 2003, we know that the human genome is less than 25,000 genes. But thanks to the Human Microbiome Project, we know that our bacterial symbionts contribute about 8 million genes to the mix. The second phase of the Human Microbiome Project is now trying to find out what those genes are doing. So that'll be interesting.
It's not 3 to 1 anymore.
Not with the genes, no. Another thing the Human Genome Project taught us is that my DNA is 99% the same as your DNA. It's that 1% that makes the difference. The Human Microbiome Project tells us that my gut bacteria may only be 10% the same as yours. So is it possible that you can be more precisely identified by looking at your gut bacteria than by looking at your own genes? Well, a recent CSI episode thinks so. They identified a killer by looking at the unique bacteria they left behind on his computer mouse. So maybe not ready for the real world, but definitely ready for prime time. CSI. I have 2 stories to share with you about the human gut microbiome. First, I want to introduce you to my friend Sandra Clifton, who was an assistant director at the Genome Institute. And she worked with a scientist there named Jeffrey Gordon, also of Washington University. Jeffrey Gordon's lab looked at the human microbiome, and they found that there's a relationship between the biome and obesity. Now, obesity always gets my attention. Because one of these days, I want to be able to sit in front of the TV and watch Cosmos or CSI, eat whatever I want, not get fat. I'm not alone in that, am I? Before I tell you about the obesity story, I have to introduce germ-free mice because they're a big part of it. Germ-free mice are born by cesarean section, they're raised in a sterile environment, and they never have an opportunity to develop a microbiome of their own. Another characteristic of these germ-free mice is that they're always lean. Even when you feed them a lot of food, they're still lean. They simply can't digest all of the food that they ingest. Back to Jeffrey Gordon, he found that the microbiome would change if you gave a high-fat diet either to mice or to humans. The species would shift a little bit in what we can call the obese microbiome. And when he transplanted this obese microbiome into germ-free mice, the germ-free mice got fat. Could it be that our gut microbiome is what makes us fat? What if we had a lean microbiome? Then could we lose weight? I'm going to come back to that, but first let me tell you about the second story about the human gut microbiome. Just as in any complex ecosystem, when you disturb it, problems occur. Lately in hospitals, there's been an increasing incidence of this condition, CDI, Clostridium difficile infection, also called C. diff. Now, C. diff are bacteria that often are part of your normal microbiome, but they're held in check by other bacterial partners that are more numerous. The problem arises when you take broad-spectrum antibiotics, and the antibiotics devastate that normal gut microbiome. Now the C. diff are freed from competition, and they produce toxins that cause deadly diarrhea. So how do you treat this? It's hard to treat it with antibiotics because that's what caused it, right? In 2011, a new treatment was tried with a patient who'd been taking a vancomycin as an antibiotic for months and still suffered from pretty severe CDI infection. This new treatment is called FMT, fecal microbiota transplant. I call it poop soup. I did warn you this topic might come up, right? So feces were taken from a normal, healthy donor, and they were given to the patient. The idea was that these normal microbiota would compete with the C. diff and alleviate the symptoms. And in fact, the patient was better after about 24 hours after the transplant. Some people think of the gut microbiome as an organ. Collectively, it weighs about 2.5 pounds. In comparison, the human brain weighs about 3 pounds. Now, the Egyptians used to do weird things with the human brain. When they mummified a corpse, they would take out all the important organs, and they would preserve those organs, each one in a different container, except for the brain. The brain, they would stick a tool up the nostril and mash it around a little bit till it was liquid enough they could just pour it right out the nose. They thought that the brain produced mucus, and who needs that in the afterlife? They just didn't understand the true function of the brain. We're just now beginning to understand the true function of the human gut microbiome. I wonder if every time we take that broad spectrum antibiotic, if we are treating our microbiome the same way the Egyptians treated the brain. Okay, let's go back to the obese microbiome because I know you want to go there. My friend Sandy retired about a year ago. Since then, she's taken several trips, including a month-long trip to Tuscany. She loves to travel. And if you look at this photo of Sandy here, you can see that she weighs about 100 pounds less than I do, give or take a couple pounds. So I floated this idea past her. I said, Sandy, you can make a lot of money for travel by producing and marketing your own poop soup. Imagine this as a lean microbiota in a fecal transplant. Don't judge me. I know some of you have already gone there based on these two stories I've just told you. But Sandy said no. It wasn't that she was a bad friend. She went on to tell me about Jeffrey Gordon's more recent studies that had to deal with identical twins. Identical twins have the same human genes and pretty much the same environment, but one of the twins in the study was obese and the other one was lean. So could the difference be in their gut microbiota? So we took the microbiota from the obese twin and put it in a germ-free mouse, and the mouse got fat. And then he took the microbiome from the lean twin, put it in the germ-free mouse, and the mouse stayed lean. And then he mixed the two mice together in the same cage. They immediately started to share their microbiota. I won't go into how they did that, but if you invite me to a party, I'm just liable to tell you.
I'm sure we can figure it out for ourselves, though.
What they found was that the lean microbiome took over. It was more diverse, and it took over the fat microbiome, and the obese mice lost weight. Now, scientists are always skeptical of their own results. So one of the researchers pointed out that these mice had a pretty healthy diet. And sometimes obese humans don't have a very healthy diet. So they decided to come up with a new food for the mice, high in saturated fat, low in vegetable material. They gave that to both of the mice, and the fat mouse stayed fat. Their conclusion was that eating a healthy diet encourages a lean microbiome, but eating a diet high in saturated fat does not. So after all of this science, what we learned is that eating a healthy diet is the best way to lose weight. Who knew? So we started this evening talking about Star Wars and the endosymbiotic midichlorians created by George Lucas. An Australian scientist, Nathan Lowe, working in Italy with some colleagues, found a new endosymbiont. It was in the mitochondrion of a tick. And they were so excited to find an endosymbiont inside the ultimate endosymbiont that the only thing they could think of was to name it after the fictional endosymbiont and name it midichlorian, mitochondriae. So they wrote to George Lucas and asked permission to do this, and George Lucas said yes. So that's how we came to share the planet with the real midichlorians. Now, I haven't been a research scientist for much of my life, but I have been able to associate with a lot of them. Either as a publisher or as an educator, I've enjoyed sharing stories about them. Stories that show the interface between science and art. Stories about Carl Sagan, who looked to the cosmos for discovery, and Lynn Margulis, who simply looked outside the box, and she found it. Stories about Sandra Clifton, George Weinstock, Jeffrey Gordon, who are giving us a new sense of self. You are not alone here tonight. You're part of a complex ecosystem that includes trillions of bacterial partners with whom you share a common ground. Those bacterial symbionts make you part of who you are. So I've enjoyed sharing these stories with you tonight. Thank you for letting me do that. And thank you to your endosymbionts. As always, may the force be with you. Thank you.